The experiment we conducted was testing the antimicrobial properties of a bio-nano-composite polylactic acid electrospun fabric impregnated with copper nanoparticles. Copper is well known for its antimicrobial capabilities and has been used for these properties for many years. With the results of these experiments, we hope to produce biodegradable and safer mask inserts for N95s that use this copper electrospun fabric to help trap bacteria. We used two different organisms to test the inhibition ability of this electrospun fabric. They were a gram-positive Staph aureus and gram-negative Pseudomonas aeruginosa. These were chosen because they're common hospital-acquired infectious agents, causing things such as MRSA and pneumonia. So these discs were electrospun, which is taking high voltage electric current and using it while forcing a polymer solution through a needle onto a collector, as you can see in figure 1. This produces small fibers, and these small fibers help increase surface area on the fabric. The fabric was tested by inoculation of the discs of the fabric in a 48 well plate, then incubated, then plated on TS trypticase soy agar and incubated overnight, and then read using CBN software that corresponded with the spiral biotech autoplater used for plating. As you can see in figure 2, this is an example of our 48 well plate. The leftmost column with the numbers means 1.5 syringe, 1 syringe, and 0.5 syringe. This is just a distinction used, and the differences between this is the time it took to make these fabrics. WO stands for without copper, and W stands for with copper. And as you can see, the leftmost column is our controls that were inoculated with no bacteria. And beside them were in triplicate our test samples inoculated with bacteria. Beside it in figure 3, you can see an example of our results collected. And these results were then taken and put into our graphs that you can see in figures 4 and 5. We only had one material that was capable of significant inhibition, and the significant inhibition was 2 log reduction as per EPA standards. And you can see that with the 1.5 syringe material, but only with Staph aureus instead of Pseudomonas. There was no noticeable inhibition of Pseudomonas with any material, and any material with the Staph other than 1.5 showed slight inhibitions on some, but nothing in the realm of significance. You can see in figure 6 that, again, there's the only 2 that have significant inhibition. Moving forward, knowing that there's only one fabric out of this set of samples that produce significant inhibition, we will test other processes of producing similar fabrics, one that is a force spun fabric and the other is one made of wood pulp. These will both be spun and impregnated with copper and then tested in similar methods to decide how to move forward and what fabrics should be used to make safer and more effective masks.